This is the first of 2 lectures on non-Mendelian genetics. So Mendelian genetics is the basic understanding that one gene is the only gene involved in a particular characteristic, and there are 2 alleles or 2 versions of it, and one is dominant and one is recessive. Now we're going to start talking about much more complicated types of genetics. And these are much more relevant to humans. So these are the kinds of genetic inheritance we will see if we're looking at people. So this lecture, we're going to talk about codominance, incomplete dominance, incomplete penetrance. Boy, those 3 words might get confusing. Pleiotropy and polygenic phenotypes. And the next lecture, we'll talk about epistasis, multifactorial traits, X-linked traits, and mitochondrial inheritance. And just to make sure that we're completely clear, these are not the only ways genes work. This is just some of the more common ways that genes work in complex organisms like humans, most animals, and most plants. Let's be honest, the peas, I've mentioned this before, are very, very simple genetically speaking. So we're going to start with codominance. And codominance, it's pretty rare, but it's something that you see in humans in our blood type. And this is a situation, in codominance, you have 2 different alleles that are both expressed and can both be observed. So let's back up and go over a little bit about the ABO blood system in humans. There's one gene that determines the identity of an antigen on the surface of the red blood cell. Remember, an antigen is something that can cause an immune response. For this gene, there are 3 alleles. The type A allele produces type A antigens. Type B allele produces type B antigens. And the O allele does not produce any antigens. So, if you have the type A antigen on your red blood cells, you will react to the type B antibody, which means that if you need a blood donation, you can receive a donation from type A or type O blood, but not B or AB. If you have type B blood, you make antigens to type A antibody, which means if you need a blood donation, you can receive B or O, but if you get A or AB, you're going to have an immune response. And it can be a very severe one, and these can end up being fatal. If you have type AB blood, this means that you have one copy of your gene that has the allele to make the type A antigen. The other copy of the gene has the allele to make the type B antigen. And you won't make an antibody to type A or B. So you can receive blood from anybody. Type O blood does not make type A or type B antigen. So it makes antibodies. If you have type O blood, you can make antibodies to A, B, and AB. So you can only receive from type O, but you can donate to everybody. So people with type O blood are considered universal blood donors. Now, There's a lot of information on this slide looking at the way the alleles line up, blood types line up. So over here on the left, there's a Punnett square. It's not a typical Punnett square. It's showing you all of the possible allele combinations. So depending on your mother's blood type, you can get A, B, or O from her. And depending on your father's blood type, the same. And that results in all of these combinations. And these combinations can give you one of, well, 4 blood types. If you've got 2 A's or an A and an O, you get type A blood. If you have an A and a B, you get AB blood. 2 B's or a B and an O, type B blood. In order to get O, you have to have 2 O alleles. So, O is recessive to both type A and type B, but A and B are co-dominant. You see both of those characteristics in the blood cells. Incomplete dominance is when you have 2 alleles that produce what's called a blended phenotype. I've been using the words characters and traits. The scientific word is phenotype, and it's the physical presentation of your alleles, or what variations of genes you have. There are relatively few traits in humans that are controlled by a single gene. So most of the time, people who talk about incomplete dominance like to show pictures of certain types of flowers that have an allele to produce red flowers and an allele to produce white flowers. And if they have one copy of each, they make pink flowers. But there is a condition in humans called familial hypercholesteremia. Familial, that means it runs in family. Hyper is excess or more or over. Cholesterol, well, that's just cholesterol. And the emia, that refers to your blood. So if you break the word down, it literally says runs in families, excess cholesterol in the blood. And this is a genetic condition. So if you have 2 mutated alleles for the familial hypercholesteremia, your liver cells do not produce cholesterol receptors. So the liver is responsible for reducing blood cholesterol. It takes it out of the bloodstream. If you have these 2, if you are homozygous for the familial hypercholesteremia alleles, your blood cholesterol can be over 4 times what is considered a healthy cholesterol or a normal cholesterol. And this will be the case regardless of your lifestyle. It's going to be high. If you have 1 copy of the familial hypercholesteremia allele and 1 copy of the normal chlorine receptor allele, you'll have half as many cholesterol receptors in the liver, and you'll still have an elevated cholesterol, but it is not as severe, and it responds well to medications. It also only usually shows up in adults. About 1 out of every 250 individuals has at least 1 familial hypercholesteremia allele. So it's Not incredibly common, but it does occur and it can be a serious health problem for people. There's also this concept called incomplete penetrance. With incomplete penetrance, there's a dominant allele, but it doesn't always give you the dominant phenotype. There's often a reduced version of the phenotype. Or you might even see the recessive phenotype. We use this term in situations where there's most likely other genes involved and or there's environmental factors involved, but we may not necessarily understand what those are yet. So it's not like there's a specific thing that happens that causes this, but it's something you might see. If you start thinking about genetics and looking at other people, it is something to be aware of. Especially if you start trying to do the, well, my partner has this phenotype and I have that phenotype, and so our kid should have this other phenotype, but they don't. So were they mixed at birth? There's so much else that could be involved, and incomplete penetrance is one of them. One of many reasons that you might be confused. Pleiotropy! Okay, so pleiotropy is when one gene affects multiple phenotypes. I have an example in humans, the cystic fibrosis gene. If you have an allele for cystic fibrosis, you're going to have problems with your lungs. This is what people think of when you think of cystic fibrosis. You think of a lot of mucus building up in the lungs. Gross. And it makes it kind of hard to breathe. But cystic fibrosis also causes problems with the digestive tract. So we've got those two systems not working correctly. It causes incredibly salty tasting sweat. So there's something else. It's a phenotype that shows up and a few other things. This is also why cats and a lot of dogs who are white are more likely to be deaf. One of the genes that can cause white fur also causes degeneration of structures in the ear. These cats are also more likely to have blue eyes. Now, the deafness comes when the cat is completely white and most of the time has blue eyes. There are other genes that involve white fur. There are other genes that produce blue eyes that do not affect hearing. So it's only one of many genes that influence coat color in cats that also goes along with deafness. This is what we call a polygenic phenotype. This is the most common mechanism of phenotypic diversity in us. And this occurs when multiple genes affect one phenotype. So I've got two examples here where it's very, very easy to see this polygenic phenotype. When we look at the top, look at that wide range of skin colors. There's not one gene. There's over 100 genes that are known to affect skin color. So you can have a wide range of skin from porcelain white to rich mahogany, and all of the diversity in between. We call this diversity where there's not a real clear line between the different categories. You know, where do you draw the line in this image between someone who's white and someone who's black? I'd be willing to bet a lot of us would draw the line at different places. And if you look at real people, it's even more confusing. Did I mention that I call this continuous variation? I don't think I did. This is continuous variation. Where does blonde hair stop and brown hair begin? These variations, if you map them out, often look like what we call a bell curve. And you can really see this in the bottom image. This is height. And height also has a lot of different genes involved. And the example here shows you these male college students from the 1914 who ranged in height from 4'10", I'm sure he got teased about that, poor kid, all the way up to 6'2". That's a huge difference. But if you notice, when they line them up like this, the shape is almost like a bell. Most of the guys are in the middle, between, let's say, 5'5" and 5'11". If you get beyond that on either end, you get much fewer individuals in each of those categories. So, with continuous variation, with a polygenic phenotype, most individuals are going to have kind of a middle trait, and a fewer individuals will have the extreme traits. The average height for American women is about 5'4". Most women are going to be within a few inches of this height. But the further away from this value, the fewer women will be at that height. So if you see a woman who's 5'6", 5'7", you kind of think, oh, they're on the tall end of normal. But if you see a woman over 5'9", she stands out. She's much, much taller than you expect. Women who are 5'1", 5'2", on the short end of the normal curve. If you get under 5 foot, then you start noticing that they are really, really short. And there aren't very many in those extreme categories. Now, the data that I just gave you, I said American women because height varies between countries. Some of it is because nutrition plays a role. Some of it is because smoking popularity plays a role. Exposure to cigarette smoke, a study done looking at boys, found that it took an inch off of their height. But there's also some genetics involved. Some countries naturally have taller people. They've got alleles in their gene pool for being tall, like the Netherlands. And other places have genes for being shorter, or they have alleles for being shorter as a major part of their gene pool. Okay, so that's some of the ways that genetics works beyond Mendel.